Gastrulation, Wikipedia audio. Gastrulation is a phase early in the embryonic development of most animals, during which the single-layered blastula is reorganized into a multilayered structure known as the gastrula. Before gastrulation, the embryo is a continuous epithelial sheet of cells. By the end of gastrulation, the embryo has begun differentiation to establish distinct cell lineages, set up the basic axes of the body, and internalized one or more cell types including the prospective gut. In triploblastic organisms the gastrula is trilaminar. These three germ layers are known as the ectoderm, mesoderm, and endoderm. In diploblastic organisms, such as Cnidaria and Ctenophora, the gastrula has only ectoderm and endoderm. The two layers are also sometimes referred to as the hypoblast and epiblast. Gastrulation takes place after cleavage and the formation of the blastula. Gastrulation is followed by organogenesis, when individual organs develop within the newly formed germ layers. Each layer gives rise to specific tissues and organs in the developing embryo. The ectoderm gives rise to epidermis, the nervous system, and to the neural crest in vertebrates. The endoderm gives rise to the epithelium of the digestive system and respiratory system, and organs associated with the digestive system, such as the liver and pancreas. The mesoderm gives rise to many cell types such as muscle, bone, and connective tissue. In vertebrates, mesoderm derivatives include the notochord, the heart, blood, and blood vessels, the cartilage of the ribs and vertebrae, and the dermis. Following gastrulation, cells in the body are either organized into sheets of connected cells, or as a mesh of isolated cells, such as mesenchyme. Classic model systems for understanding gastrulation. The molecular mechanism and timing of gastrulation is different in different organisms. However, some common features of gastrulation across triploblastic organisms include a change in the topological structure of the embryo, from a simply connected surface to a non-simply connected surface, the differentiation of cells into one of three types, and the digestive function of a large number of endodermal cells. Louis Wolpert, pioneering developmental biologist in the field, has been credited for noting that it is not birth, marriage, or death, but gastrulation which is truly the most important time in your life. Mollusk, sea urchin, frog, chicken. The terms gastrula and gastrulation were coined by Ernst Haeckel, in his 1872 work Biology of Calcareous Sponges. Although gastrulation patterns exhibit enormous variation throughout the animal kingdom, they are unified by the 5 basic types of cell movements that occur during gastrulation: 1 invagination, 2 involution, 3 angryzion, 4 delamination, 5 epiboly. Gastrulation is highly variable across the animal kingdom but has underlying similarities. Gastrulation has been studied in many animals, but some models have been used for longer than others. Furthermore, it is easier to study development in animals that develop outside the mother. Animals whose gastrulation is understood in the greatest detail include: the distinction between protostomes and deuterostomes is based on the direction in which the mouth develops in relation to the blastopore. Protostome derives from the Greek word protostoma meaning first mouth, whereas deuterostome's etymology is second mouth from the words second and mouth. The major distinctions between deuterostomes and protostomes are found in embryonic development. Mouth/anus. In protostome development, the first opening in development, the blastopore, becomes the animal's mouth. In deuterostome development, the blastopore becomes the animal's anus. Sea urchins' euechinoidea have been an important model system in developmental biology since the 19th century. Their gastrulation is often considered the archetype for invertebrate deuterostomies. Sea urchins exhibit highly stereotyped cleavage patterns and cell fates. Maternally deposited mRNAs establish the organizing center of the sea urchin embryo. Canonical WNT and delta notch signaling progressively segregate progressive endoderm and mesoderm. Protostomes versus deuterostomies. In euechinoids, the first cells to internalize are the primary mesenchyme cells, which have a skeletogenic fate, which ingress during the blastula stage. Gastrulation internalization of the prospective endoderm and non-skeletogenic mesoderm begins shortly thereafter with invagination and other cell rearrangements, the vegetal pole, which contribute approximately 30% to the final archenteron length. The gut's final length depends on cell rearrangements within the archenteron. Tailless amphibians are a classic model system for gastrulation. The sperm contributes one of the two mitotic asters needed to complete first cleavage. The sperm can enter anywhere in the animal half of the egg, but its exact point of entry will break the egg's radial symmetry by organizing the cytoskeleton. Prior to first cleavage, the egg's cortex rotates relative to the internal cytoplasm by the coordinated action of microtubules, in a process known as cortical rotation. This displacement brings maternally loaded determinants of cell fate from the equatorial cytoplasm and vegetal cortex into contact, and together these determinants set up the organizer. Thus, the area on the vegetal side opposite the sperm entry point will become the organizer. Hilda Mangold, working in the lab of Hans Spemann, demonstrated that this special organizer of the embryo is necessary and sufficient to induce gastrulation. Specification of endoderm depends on rearrangement of maternally deposited determinants, leading to nuclearization of beta-catenin. Mesoderm is induced by signaling from the presumptive endoderm to cells that would otherwise become ectoderm. The dorsal lip of the blastopore is the mechanical driver of gastrulation. The first sign of invagination seen in this video of frog gastrulation is the dorsal lip. In amniotes, gastrulation involves the creation of the blastopore, an opening into the archenteron. Note that the blastopore is not an opening into the blastocele, the space within the blastula, but represents a new inpocketing that pushes the existing surfaces of the blastula together. In amniotes, gastrulation occurs in the following sequence: the embryo becomes asymmetric, the primitive streak forms, cells from the epiblast at the primitive streak undergo an epithelial to mesenchymal transition and ingress at the primitive streak to form the germ layers. In preparation for gastrulation, the embryo must become asymmetric along both the proximal distal axis and the anterior-posterior axis. The proximal distal axis is formed when the cells of the embryo form the egg cylinder, which consists of the extraembryonic tissues, which give rise to structures like the placenta, at the proximal end and the epiblast at the distal end. Many signaling pathways contribute to this reorganization, including BMP, FGF, nodal, and WNT. Visceral endoderm surrounds the epiblast. The distal visceral endoderm migrates to the anterior portion of the embryo, forming the anterior visceral endoderm. This breaks anterior-posterior symmetry and is regulated by nodal signaling. Sea urchins. Symmetry breaking. The primitive streak is formed at the beginning of gastrulation and is found at the junction between the extraembryonic tissue and the epiblast on the posterior side of the embryo and the site of angryzion. Formation of the primitive streak is reliant upon nodal signaling in the collar sickle within the cells contributing to the primitive streak and BMP4 signaling from the extraembryonic tissue. Furthermore, CER1 and LEFTY1 restrict the primitive streak to the appropriate location by antagonizing nodal signaling. The region defined as the primitive streak continues to grow towards the distal tip. Germ layer determination. Cell internalization. Amphibians. Symmetry breaking 2. Germ layer determination 2. During the early stages of development, the primitive streak is the structure that will establish bilateral symmetry, determine the site of gastrulation and initiate germ layer formation. To form the streak, reptiles, birds, and mammals arrange mesenchymal cells along the prospective midline, establishing the first embryonic axis, as well as the place where cells will ingress and migrate during the process of gastrulation and germ layer formation. The primitive streak extends through this midline and creates the anteroposterior body axis, becoming the first symmetry breaking event in the embryo, and marks the beginning of gastrulation. This process involves the angryzion of mesoderm and endoderm progenitors and their migration to their ultimate position, where they will differentiate into the 3 germ layers. The localization of the cell adhesion and signaling molecule beta-catenin is critical to the proper formation of the organizer region that is responsible for initiating gastrulation. In order for the cells to move from the epithelium of the epiblast through the primitive streak to form a new layer, the cells must undergo an epithelial to mesenchymal transition to lose their epithelial characteristics, such as cell-cell adhesion. FGF signaling is necessary for proper EMT. FGFR1 is needed for the upregulation of SNAI1, which downregulates E-cadherin, causing a loss of cell adhesion. Following the EMT, the cells ingress through the primitive streak and spread out to form a new layer of cells or join existing layers. FGF8 is implicated in the process of this dispersal from the primitive streak. There have been a number of attempts to understand the processes of gastrulation using in vitro techniques in parallel and complementary to studies in embryos, usually though the use of 2D and 3D cell culture techniques using embryonic stem cells or induced pluripotent stem cells. These are associated with number of clear advantages in using tissue culture-based protocols, some of which include reducing the cost of associated in vivo work, being able to accurately apply agonists/antagonists in spatially and temporally specific manner which may technically difficult to perform during gastrulation. However, it is important to relate the observations in culture to the processes occurring in the embryo for context. Cell internalization 2. To illustrate this, the guided differentiation of mouse ESCs has resulted in generating primitive streak-like cells that display many of the characteristics of epiblast cells that traverse through the primitive streak, and human ESCs cultured on micro-patterns, treated with BMP4, can generate spatial differentiation patterns similar to the arrangement of the germ layers in the human embryo. Finally, using 3D embryoid body, and organoid-based techniques, small aggregates of mouse ESCs are able to show a number of processes of early mammalian embryo development such as symmetry breaking, polarization of gene expression, gastrulation-like movements, axial elongation, and the generation of all 3 embryonic axes. Amniotes. Overview. Symmetry breaking 3. Germ layer determination 3. Cell internalization 3. Gastrulation in vitro. Notes. Bibliography.